Very warm good morning from IHR. As per yesterday's ranking, we are this Zoom link is from the best institute in IHR. It is not just for information, those who joined and not seen that news, the only for that purpose I'm sharing this. Then respected Professor Dr. Tivika Singh, Dr. Nagesh, who joined from NBR, Dr. Veena from CTCRA, and whole lot, all the participants who joined in this online talk of AAPMHE, which has been continuing in the last 110 months without any interruption. Every month we are having one talk or sometimes two talks also. Today, I'm happy to share that today with us Dr. M. Krishna Reddy sir. Actually, this particular topic was scheduled for June on the day of retirement. Because of some urgent meeting that day, we could not take it up, and it is being postponed. And we are having today. We can have that day. Dr. Krishna Reddy sir was so busy, and today we can have little bit more discussion on this interesting topic. As all of us know, these viruses are very tricky organisms for handling because of this their frequent evolution or mutation or whatever we call. That is that what we are going to listen from Dr. Krishna Reddy sir on the topic origin and evolution of plant viruses. Before that, I will give a brief account of this today's speaker, Dr. M. Krishna Reddy sir. He did his MSc from Marathwada Agricultural University, Parbhani, and during that time you had 3 medals from that MSc, gold medal and 2 other meritorious medals for his distinguished achievements as a MSc graduate. After that, he joined Indian Agricultural Research Institute, New Delhi, and 89 he completed a PhD in virology under Dr. Anupam Verma, very well-known plant virologist. Then he did postdoctoral fellow at National Food Research Institute, Tsukuba from Japan. Coming to this employment experience, he joined at IHR in 1990 until his retirement recently in June 2022. He had a vast experience of 32 years working on plant viruses and their management in different horticultural crops. He has, for his credit, to his credit, several awards, like very recent most influential plant pathologist in Asia Pacific, awarded by Asia and Pacific Seed Association, Bangkok, Thailand. Then ICAR Award for Outstanding Interdisciplinary Team Research in Agriculture and Allied Sciences for Crop and Horticultural Sciences in 2016. And ICAR Award for Outstanding Interdisciplinary Team Research for the Biennium 2007 and 8 also. Harbhajan Singh Memorial Award for Best Paper published in Vegetable Science by Indian Society of Vegetable Sciences. Apart from this,
Singh sir,
yeah, sorry, there was some bandwidth problem I lost, and I will continue where I missed this biodata reading. In continuation, Dr. Krishna Reddy sir, he got IHR Foundation Day Special Award. I hope I am audible. Dr. Madhu Reddy madam. Am I audible? Can anybody?
Yes, now you are audible.
Audible, sir.
Okay. Then he is a member of International Committee on Taxonomy of Viruses. He is a RAC member of ICAR CPRI, and he's and apart from this, there are for several institutes. He is a member of advisory committees, and he is a member and fellow of several professional societies like AAPMHE, for which he also served as a president for 6 years, and he is fellow of Indian Virological Society, New Delhi, fellow of Indian Phytopathological Society, New Delhi, and fellow of Confederation of Horticultural Association of India. To his credit, more than 175 research papers, 11 book chapters, and five technical bulletins are there. He mentored one postdoctoral fellow, 9 PhDs, and 6 MSc students. And during his tenure, he garnered around 70 crores worth external funding from different agencies from outside India as well as in India, from India, with a total fund of around 70 crores. One of the most worthy thing from institute, if you recall, he has developed maximum facilities required for the plant protection, like functional, including functional TEM, SEM, and other facilities. And he also led the team for the issue of phytosanitary certification, which is very, very important for all horticultural crops. That is in brief about Dr. Krishna Reddy sir, today's speaker. With this brief introduction, I request Dr. Krishna Reddy sir to go ahead with his presentation on origin and evolution of plant viruses. Thank you. Over to Dr. Krishna Reddy sir.
Thank you, Dr. Sridhar. Good morning to all. I wanted to make people should understand about viruses because we have normally we heard there is no control, this is cut and burn only. To say that this is not true with that. That is the reason why I chose this topic to introduce and make understand that why it is necessary. Sridhar.
Hi, sir. You can share the slides, sir.
Since sharing you allow, it is host disabled. It is coming.
Yeah. Okay. Now you do, sir. I gave sharing permission.
I think you are able to see now.
Sir, now it's clear.
Let me see the origin and evolution of plant viruses and the evolution and emergence. If you see the life of origin, how life is mentioned, among the living, I consider virus as a living. Let us, when we talk, we will come. And this is the abundant form in the universe. It is the living ones. It is also evident that viruses played a major role in driving the cellular evolution. Then virus played a primary and critical role in evolution of now in the mainstream biology, the tree of life, bacteria, archaea, and eukaryote are included. Being a particles, having some non-living entity. This is if you realize the life, the first plant virus symptom description, who wrote in her poem that the yellow leaf of Epitorium, describing a poem, and in 18th century, Dutch tulip bulbs, where the variegated bulbs are the virus infected, are given as a gift to the princess, and that is how the oldest record of viruses known. The Russian botanist Ivanovsky was the first to pass through this tobacco-infected samples through porcelain filter and said that it can pass through the microbial filters, which retains the microbes. It is not an organism, it will be a toxin. But in the 1898, Bezernik was the first to show the filterable agent is not a toxin, but it is a microbe which can. And then he called it as a contagium vivum fluidum, is a living fluid of infectious nature. But in 1935 is the first Stanley fluid, not the fluid nature, it has a definite cellular link with the microorganisms. Crystallization of these particles and also seen the shape as a rod-shaped virus particle. And the first electron micrograph of virus was shown in 1939, that is the TME. And again, Baden during that time is defined as an obligate parasite. Virus, not other virus. Later on, there are several viruses which are seen through electron microscopy and different shapes and sizes have come. If you see the dictionary of virology, it is defined infectious units. Of viruses now has expanded because the functions of viruses has changed and people understood more about viruses. And this was observed in some animal cells, multiplying in the form of their genetic material, unable to grow and undergo binary fission and divide of Lippmann system. This was the definition of partly based on the animal. And in protozoa and in plants, infectious, potentially pathogenic nucleoproteinic entities. That is the change. Intracellular is removed because there are intercellular and intracellular presence of virus particles. Then they have been given as a nucleic acids and are produced from the genetic. It is the only link, the divide of Lippmann system and binary fission are the only points which virus do not have. There it is different. This is now considered as a link between living and non-living. That way it will be evolving from a non-living to living in the form of genetic information, having the characters of both. That is, this could be the primitive cellular entities which has from there evolved. See, if you see the common ancestral origin, if you called bacteria, archaea, and eukaryota, but there are ancient cells which theories and hypothesis of origin of virus. There are three theories are proposed. One is the pre-cellular theory, wherein the virus particles existed in the cell, the nucleic acid existed in the cell, and then with the host protein machinery, they have a protein coat then made along with the then in the modern cell theory, they have been an independent of a cell and it can infect the cell and survive in the cell. In the regressive or reduction hypothesis, where the remnants of cellular organisms are the ones which develop in there is a cell with some nucleic acid has gone as a reductions in the functions that is led to the virus development. The reductive cell as viruses. Whereas the escape or endogenous theory, it says that the procell has this virus nucleic acid, but it is maintained they have been evolved as a separate entities. That is the theories of this, which has led to it. But now the procell from the ancient time and the modern cell concept today, now there is a link, the hybrid chimeric model recruitment of cellular proteins for capsid proteins to escape from cells is the one which is the confirmation that the chimeric nucleic acid, its proteins are derived from host machineries. Model wherein they say that the hypothesis ancient cells coexisted with the ancient virus cells and the modern day viruses evolved by in the cell process multiplication utilization their machinery host cell systems. So the living cells. That is why they say that this is the both has in association evolved from the ancient time. And if you see the now current molecular approaches using genomic tools, the phylogenomic analysis has revealed that they share certain genes which are common between archaea, eukaryota, bacteria, and viruses. And viruses becomes the base of it, followed by bacteria, then archaea, then eukaryota. So that is how the evolutionary process they are connecting. And if you see the origin of viruses and having proteins and nucleic acids as analyzed independently, separate genes when analyzed, it is well connected some of the polyproteins and the proteins which are necessary for a living form to multiply, and they share common these things. In that respect, virus shares certain genes with eukaryota, archaea, and bacteria. So that plays the now link that it is a link between the living form to non-living form. And see what are the, if you see the concept of now hologenome, wherein the concept of evolution postulates that the holobiont, host plus symbionts, what we consider as the earlier only with respect to bacteria association, we say symbiotic association, and in mycorrhizal association with symbiotic. Like similarly, now with hologenome concept, the level of selection in evolution is changed. The term holobiont was first introduced by Murgils to describe that host and its endosymbiont. But Shower in 2002 described that choral symbiont is also includes the bacteria, archaea, protists, and viruses. There are 4 components. Normally, it is the hologenome concept is associated only with microbes, but now it is been reclassified that the advances in the genomic understanding led that there is an association of viruses as well as a holobiont. The multicellular organisms can no longer considered as individuals. We all think that the human being means is an individual. But in human itself, the gut system are on the surface of the scale. Several microbes exist which are living along with that. In addition to now, the concept is bacteria, the viruses are also present in the system. When viruses expresses into some form, then we consider as a disease, and then that is the viral diseases come. But there are certain viruses which without expression, they carried in the individuals or cells. So that is how now the holobiont concept says that viruses are integral part of the living system or in a living cell. So now the fundamental framework understanding interaction between host and microbes is in case of health and disease, and evolution makes an important aspect that there are the multicellular organisms and there are single cell organisms, and in each cell is not a cell is a mixture of genomes, both microbes as well as we know even that the mitochondria is a independent evolved and it is carried in the cell. Similarly, the virus is also existing in cells.
Excellent, sir.
The first description of virus also is only from plant only it is studied. Subsequently, animal and bacterial viruses were all introduced. So the majority known plant viruses are RNA viruses. The origin of RNA virus are thought came directly from the ancient RNA world, the time before cellular life began, where RNA replicated itself without a DNA phase. Then subsequently, there is a DNA being evolved from the RNA, and both RNA and DNA are evolved. But specifically the hallmark of genes is that across many families of viruses, such as RNA-dependent RNA polymerase, are analyzed to similarity across wide range of animal and plant RNA viruses with positive, negative, and double-strandedness genomes. In every viral genome, the RNA-dependent RNA polymerase exists irrespective of RNA or DNA. And with hallmark genes are shared between selfish genetic elements such as plasmids and transposons, suggesting that viruses have a deep lineage with some suggest dates to prove cellular time. And indeed, example viral elements being incorporated into the host cell genome can be found across all tree of life. The best examples, even in plants, we have a BATNA viruses which are integrated in the host genome. In humans also, the human many virals are integrated in host. For example, you have what we see jaundice. Some of the forms, hepatitis C, A, B, C. A is the one which is silently, it won't show the symptom, but it carried in the humans. And it can pass through from a parental to the offspring, and it is carried several generations. Genetically, it is carried and continued. So this is how some of the viral genes are incorporated in the host and existing in the. Similarly, in case of human, animals also has certain viruses which are integrated in the host, but expressed only during certain stress conditions. If you see the lifestyles of plant viruses, like we have a lifestyle, same way have 4 different lifestyles. One is the persistent, which can be there for continuously. Then acute, one which come at certain time and go again. Then chronic, continuously, and then endogenous. They are present in the inside, but they won't express any time. Some of the virus may be able to change from one lifestyle to another, particularly between the acute and the chronic lifestyles, and other lifestyle change probably occur but are a rare phenomenon. The persistent plant viruses have a generally asymptomatic, although lack of uninfected plants in most cases make this difficult to fully assess. They are transmitted vertically via gametes, but not horizontally. Insects are not involved in transmitting these viruses. Some of the best known examples are the endoRNA viruses and the cryptic viruses which are present. They do not move between cells in the plants, but even though through grafts, rather they found in every cell, including the meristem. And in acute plant viruses, in contrast to persistent viruses, have a difficult life cycle, different life cycle. They most frequently transmitted horizontally, and they may not be transmitted vertically, but in some hosts they transmit vertically, and vertical transmission can be through embryos or gametes. Most of the viruses, and we have a common notion when several viruses now are infecting across several kingdoms. And recently, when we have seen this, the smallpox time to today corona, where we say that these viruses are evolving. But in zoonotic diseases, viruses come from animals to the humans. That is common. And the emerging new viruses are the shift of viruses from animals to the humans or vertebrates, among vertebrate circulation. Therefore, the virus changes between animals and human is very common, and they share a large part of their relationship. Whereas invertebrates and vertebrates has a very little exchange, and between invertebrates and the plants, again, there is a common little sharing, but there is no share between plant and the vertebrate viruses. So therefore, till date, there are reports say that some viruses are found in human beings. They are found, but they are not being shown till date an association of a pathogenicity, means a disease-causing things has not been demonstrated till date in animals by the plant viruses. So therefore, the other aspect of this is also because of the leaf, the systems which they are included, mainly the cell difference. The animal cells and plant cells primarily different in terms of their structure. This led to the virus passing and transmission, difficult in case of between vertebrates and the plants. And let us see why we have so much in today. Viruses are so much in case of plants. I don't think that as a virologist, I am highlighting that viruses are more, but I am showing with the scientific data. See, one of the reason is the international trade and travel, long distance transport of planting material, then climate change, increase in frequent unusual weather events, change in the distribution of vectors are the primary source for viruses to spread, and the agriculture cropping patterns we changed from intensity and acreage, non-traditional area to expansion, large-scale monoculture and continuous cropping, and changes in the genetic composition of host population. We breed and bring a lot of change in the population of host, change in the ecology and genetic composition of the vector, which is again very similar to the virus, and host pathogen evolution, mutations, recombinations, and evolution of biotypes due to biotic pressure in the insects. If you see, among the pathogenic forms in the plants, now currently 40%, more than 40% are the viruses, and 32% are the fungus, and 10% is the bacteria, and 11% is the phytoplasma, and 4% is the virides. Among these pathogens, you can see that the range and the increase in this number is not because of these were not known earlier. With the new approaches and new technologies are bringing in more number of virus identification. So therefore, that is how it is led to the increasing in the population. Accordingly, if you see as per the taxonomy of viruses, among the plant viruses, we have 4 groups of viruses. One is basically based on genome, DNA and RNA. In DNA, we have a double stranded and a single stranded. And in RNA, again, double stranded and single stranded. In single stranded, again, we have negative sense RNA and positive sense RNA. So the positive sense RNA are the major contributing more than 50%, followed by the single stranded DNA viruses, and then the double stranded RNA. So this is how the different shapes and sizes one side were classified. Current classification is based on the genome part of the viruses. If you understand the viruses, the life cycle of a virus is take from an infected plant, it will multiply and pick by either insects or through seed, through pollen or seed multiplication. Then it will become to the seed is infected or a planting material get infected. From there again virus will come for the expression. In all this process, the critical aspect is the insect vectors. Unlike in human viruses, insects plays 40% only, 60% are played by other factors, physical and the water. Whereas in plant viruses, 80% of the role is played by insects and 10% is played by fungi and nematodes, and 5% is through seed, and 5% is the contact without involving any agents. This is how the spread and survival of the plant viruses exist. And just I have taken few examples to say the evolutionary pattern of the plant viruses. Unlike, see, you all been observing, the whitefly transmitted viruses has increased enormously. Earlier, the largest number of plant viruses are the potyviruses, the aphid transmitted potyviruses. Today, it is overtaken by the whitefly transmitted bigomoviruses, is the single stranded DNA viruses, which are the now #1 group of plant viruses, which are among the plant viruses. And if you see the number of hosts, it was earlier a few hosts. We know okra, yellow bean, and legumes and yellow mosaic, the earlier ones. But today, you name any plant, bigomovirus is infecting. That means it is conquering to every host plant and changing the behavior of the host. And it is now also seen that these variations are also depending on the environmental interaction and the insect vector modification. They are becoming very potential pathogens throughout the world. And if you see the evolutionarily how these DNA viruses are existing, initially it was seen that the pre-mastery virus, means the ones which infect serial Gemini viruses are the pre-existing, and then from that it is evolved to become two types of particles, the single stranded DNA and the single component DNA, monopartite, and the double component bipartite ones. From there, the helper components have been evolved. This is how in Geminis. And in addition, these changes also brought to the DNA viruses prevalent in others like the bunchy top and the Faba necrotic yellows, and coconut foliar decay. All these are all single stranded DNA evolved in different directions and existed. But today, with the information generated with the molecular tools, there are now more than 11 groups of viruses have been identified, which have a single stranded DNA, which is a Geminiviridae family. And if you see, among the largest in the Geminiviridae is the bigomovirus, as I was mentioning. And in this evolutionary, again, the ancestral one is the single stranded DNA, which is split into a double stranded DNA. We have again double stranded DNA variations. In the new world, slight variation, certain genes are not there, whereas in the old world, certain genes are differ from that of a new world ones. From the ancient monopartite, the monopartite has led to bipartite groups. And again, with the bipartite, there is split and splices of DNA with the common sharing intergenic region has developed the satellite DNA molecules. There are now today 3 different satellite molecules. One is the alpha satellite, another is the beta satellite, another one is the delta satellite. in bigomyrises as a particles, if you see, it looks geminate particle, but the genome is a single, means it is monopartite. If it is divided, it is bipartite. The supporting the satellite molecules are the alpha, beta, and delta. So this is how, if you see genome organization, which shares the common between the monopartite and bipartite, and there is a common region between the satellites. This is the one sharing point between the helper components and the satellite molecules. So this is how initially we have identified, we know that only bigomyrises are there as a monopartite or bipartite. But today with the advancement, the first described what the beta satellite, then alpha satellite, now delta satellites. So that is how the evolutionary. And if you see the bipart nature of the virus are less prevalent now compared to the monopartite, with the satellites as increasing, which is leading to the spread and enhanced host range and infection. Among these, I would like to take one example is the tomato leaf curl New Delhi virus is a bipartite virus, but it is known first recorded in India. Don't think that New Delhi means it is from the New Delhi, but where the virus first reported, that name is given in the virus nomenclature. Once that is noticed, and it was more prevalent in northern part of India, now it is spread to the Eastern Asia, then the Middle East, now Europe. In Europe, it is spreading very fast, and it is spread is and very enormous, especially in cucurbits, followed by solanaceous crops. This virus is the one which infects a range of host plants, from annuals to the perennials, and from there is no specific host which you say this is only specific to this. Now this is a polyphagous virus spreading worldwide and concern for many. And not only that, this virus will also have a seed-borne nature, which we will deal in subsequently. Earlier in the before 90s, we have some reports of viruses transmitted by grasshoppers and some other insects, or 2-3 insects, which is because of lack of identity of the virus. It was only named based on symptom, but not the exact virus identity. Today, we have a definite tools to identify virus and the insects, and there is a definite relation exists as evolutionary mechanism for transmission of viruses. One example is in a same whitefly species, there are 2 different genera which transmits a single virus. Is the Creeny virus, there are 2 viruses here, tomato chlorosis virus and cucurbit yellow stunting disorder virus. Both are transmitted by the Bemisia tabaci, the tobacco whitefly, and the Trialuridis, which is a glasshouse whitefly. Both will transmit a single virus. In case where 2 different genera of same whitefly is transmitting this virus. And if you see the largest virus earlier, not now, is the partyviruses, which were existing as a monopartite. Now there is a bipartite virus also, bimovirus, where it has 2 split genomes in the partyviruses. Partyviruses earlier were majority transmitted by aphids. Now partyvirus also transmitted by whitefly as well. Insect is there. In a single genus, whitefly can be transmitted by 2. What I mentioned is Creeny virus is transmitted by whitefly, which belongs to Partiviridae, whereas partyvirus is transmitted by aphids, different groups of aphids in this. And the another group which I would like to express is the persistent and propagative virus, wherein it is earlier is a luteovirus, which is now called Swallomoviridae, wherein with the genetic information which have a genome similar nature have been grouped into Swallomoviridae. In that we have 2 virus groups, one is Sobemovirus and Polymovirus, which occur mechanically wounding, vegetative propagation, and some insect are through soil they are transmitted. But polarivirus and enormoviruses are transmitted specifically by aphids only in a persistent manner. This is the concept existed till 2 years before. But today, after 2 years, we have, these are the phloem bound viruses existing, which are phloem feeding aphids are transmitting these viruses. With the recombination or modification in the host genome, there is a recombination which leading or addition of certain amino acids. These viruses now currently are transmitted by whitefly as well. Here is an example between where the polariviruses are known to be transmitted by aphids are also can be transmitted by whitefly if the genome modification happens in the vector binding region. So this is being demonstrated in 2 viruses. One is the pepper vena yellow virus, another is the cucurbit aphid-borne yellow virus. In both the cases, modification in the genome led to the whitefly transmission. And if you see again, there is the oldest virus is the TMV, wherein we know that virus existed from centuries old and the first recorded virus. From that time now, the same group as now developing several new viruses with the recombinations. The recently now the concern for the movement of germplasm and the seed was the tomato mottle mosaic virus and tomato brown rugose fruit viruses. If you see the analysis part, and the brown rugose has uniformly evolved virus, whereas tomato mottle mosaic virus has a genome from TMV, TOMV, and pepper mottle virus, which has now evolved as a new virus, and it is now spreading in solanaceous hosts. And the cucumber mosaic virus, again, an old virus and known for a symptom for a long time and a serious problem in several hosts. But currently, why it is important? Because several epidemics throughout the world is happening, and same thing happened in India for past 5 years. What is the nature of this virus? Why I am, as we as a virologist or a pathologist, we know the mosaic symptoms observed or a filiform nature in the leaf is observed, we definitely say it is a CMV. But symptom is not the criteria today. Expression will change according to the genotype and the environment. Now we see this virus is not showing expression in the leaf, but shows the symptoms in the fruit. The fruit turns yellow or browning, and it is not showing symptoms in the plant, which is past 5 years. This is the problem in Maharashtra. We have investigated and studied and found that this is the CMV virus, which is not expressing symptoms in the plant. But if you take from the fruit and inoculate in the plant will show again symptoms. But in the natural condition, it is not showing symptoms in the plant. Same thing has happened. We observed first in the case of tomato, but last 2 years, we also observed similar case in case of chili, wherein fruit symptoms are expressed, but not in the plant. So there is a change in the behavior of the virus and the evolutionary process, and the expression is leading to the variation. Our symptom variations happens. See, any successful establishment of a virus is there is opportunities in its life. One is the generalistic viruses where they have different hosts, where more hosts like CMV and tomato leaf curl noodle virus. Their selection pressure on the virus is least, and they are able to adapt differently into different hosts and selection of unique genotypes, and then capable of infecting. When it is infects a host, there is a slight change in the genomic nature, modifications, and that modified strain or isolate will go infect to a new host or different host range. So that is the how, and this is all done by the insects. The insects relationship exists very close relationship, that is the protein-protein interaction and the stylet, wherein it picks certain isolates and inoculates into. See, insect doesn't differentiate which host to inoculate. It will go and feed in several hosts which it is feeding. When it is feeds, the virus goes. If virus is successful, it is lead to symptom development. If virus is not able to replicate and multiply, it will be lost. That becomes a dead host there and the virus is lost. So that is how in nature, several virus clones are picked and transmitted, but the success ones are only the ones which are expressed in the plant. That is how fitness exists in the natural system. If you see the transmission modes of the viruses, we have the three modes main for contact is the normal, which is a tobamoviruses, mechanical transmission by touching or sap one plant to other. So there is a less evolution in these viruses. Whereas through seed, certain viruses carried in the plant through pollen and the seed reaching to the seed embryo and then multiplied and then establishment. It is from the gametic relationship to pollen, there is a transmission of true seed transmission happens. And certain viruses are exist in the pollen, but they may not be able to establish in the seed. So the pollen carried and subsequently in the gametic phase, they are not able to establish. That is how their evolution is. So when there is a change in this type of nature, there we have a certain viruses which are again, we reach to embryo. In the reaching from a mother plant to the mesophyll cells to the meristematic cell to the reaching the embryonic cells and then developing in the embryo axis and through pollen or directly through phloem tissue. When this is the system is connected, we have two types of viruses. One are the externally born seed-borne viruses, wherein they could not establish in the reaching to the mesophyll or the cells of embryonic cells. So they lack, so therefore they cannot reach that. They are not in the internally seed-borne nature. They exist in the seed coat because their existence is on the surface. Whereas certain viruses are able to reach the embryonic cells and they exist in the embryonic cells and then through embryo to the hypocotyl, then it will establish in the plant and show the symptom. These are the true internally seed-borne viruses. The externally seed-borne are the tobamoviruses which are known earlier, now also very much existed. When we tested the different components, you find that they are detected only in the seed coat or cotyledon, but not in the embryo axis. Whereas in the internally seed-borne, which are exist in the seed coat as well as in the cotyledon and also in the embryonic axis. That is how the viral survival and evolutionary. Why I brought this information is, we know certain viruses like bean common mosaic, bean yellow mosaic, and the cucumber mosaic, and all are seed transmitted. They're truly in the seed, which were known, which are aphid transmitted. No whitefly transmitted bigomoviruses are seed transmitted till 2005. The reason being, these all bigomoviruses are phloem limited and phloem connectivity is not there with the embryonic axis. Therefore, it is not transmitting. That was the assumption we had. But today, these bigomoviruses are evolved and adapted to reach the embryonic cells, and there is now they are also internally seed-borne. That is the #1 virus which is found was the tomato yellow leaf curl bigomoviruses, first reported in Korea and subsequently in Italy and in Israel. This virus is found to be seed transmitted in tomato. Then later on, our work and Dr. Maluti work demonstrated bitter gourd, the same tomato leaf curl New Delhi virus, which is again seed transmitted, found in the seeds, and it is internally seed-borne. This is the reason why this tomato leaf curl New Delhi virus was spread from Asia, India, to the East Asia, to Thailand, other parts, and also in Europe. The movement would have been through seeds which carried this virus and established, and subsequently the ecosystem, the prevailing whitefly biotech picks these isolates and spreading there in that. So that is the evolutionary point of these bigomoviruses are seed established and now transmitting. But there is a caution to be mentioned to everybody. Seed transmission doesn't mean, that is what evolutionary point of view to speak. All the seed transmitted virus will not equally transmit in all genotypes. If in the same genotype, in the same, say for example, bitter gourd, different varieties will vary with respect to seed transmission. It is again the interaction between virus and the genotype which leads to success in transmitting. So the virus is able to evolve and transmit to the reach the embryonic. It happens in certain genotypes, but not in all the genotypes. So that is why we have to apply this concept cautiously and checking, because when we say the tomato leaf curd New Delhi virus is seed transmitted, doesn't mean that all varieties will have a seed transmission. It is genotypic and virus interaction and strain interaction which lead to the successful seed transmission. That is where we need to understand this evolutionary mechanism. And if you see the insects as a transmission modes, we have a different groups of insects, the aphids, leafhoppers, and beetles. All of these transmits primarily. There are 4 different transmission modes which we seen. The non-persistently transmitted, wherein it is a merely contamination of the stylet, which they take attachment and transmit. But this contamination, it doesn't mean that it only can pick, but there is a definite association of a protein which is stylet attaching. So that protein only, if it is present, is able to pick and then turn. Most of these viruses which are present on the mesophyll cells are picked by this type of transmission modes, mostly aphids, and they transmit in a short time, and they retain the viruses also very short time, less than 1 hour. Whereas in semi-persistently transmitted viruses, most of the whitefly and some aphid transmitted, wherein the virus is present in the phloem and also in the mesophyll, it picked, and there is again definite association of a protein involved in attaching to this, and that will be circulated only in the salivary gland, but not in the hemosyl, and it is not circulated. So that is why this variation happens in this mode. Whereas in persistently non-propagative and propagative virus, the viruses are not present in the mesophyll. They are definitely present only in the phloem, and the insects which feed only on the phloem cells are able to pick these viruses. So there is an association again. There are movement proteins which link to the phloem movement of these viruses, and there is proteins which are involved, the stylet attachment. Both are there, then only the insect is able to pick. And then these viruses have 2 hosts in their preference. One is the plant host, another is the insect host. Insect is also a host for a virus because it acquires place in the host insect body and survives in the insect body. There is a slight modification happens in the genetic structure in this, and that genetic structure modified once is host to plant, and that is how the evolution changes and the host selection process by the insect vector and the virus will play important role in these things. In all these interactions, if you see the climate is one we implicated for several things, but as an environment, virus changes, incidence, virulence, host range are determined by the insect vectors, emerging strains, and the host selection. All these are all influenced by the climate. When there is a pressure on the host metabolism and there is a change in the host physiology, there is insects which prefer in feeding or avoiding, and there is a selection pressure on the pathogen, which whether new strains modification or recombination happens, that lead to the pickup of these new strains. If any of these systems we need to study in terms of ecology and evolutionary mechanism, we should understand the ecosystem. If you see the ecosystem ecology, the impact of biotic stress are equally important for abiotic stress are equally important for a biotic stress. Changes in the land use pattern will change the dynamics of virus, And the expansion of geographical range of arthropod insects will also change the behavior of the viruses. If you see as a pathogen, viral community ecology, as a population, if you see virus study, there is a co-infection, a rapid spread, which happens lead to the recombinations, resortment, and mutations. Then which leads to the evolution of a new strains or a new viruses. And there is shift in viral ecological network, means from a host shifts from one host to another, or multiple hosts, or multiple vector selection, as we seen in one example is the polariviruses. And host insect tripartite interaction is very, very crucial for viruses. Viral evolution and adaptation will mainly depending on these interactions. Fitness of a virus host and also depends on this. If you see the manipulation of host plants, arthropod vector emerged as important components, because change in the vector orientation, behavior, settling, feeding, all this led to the infection of host more successful. And the pathways of vector multiplication is two ways. One is the direct and indirect, because direct ones is the direct attraction and change. Indirect ones are the ones which changes the physiology and the behavior of the insect modification. There are direct effects also on vector behavior for relevant for transmission. That there is enhanced attractiveness of the insect vectors for a particular virus. If you see that plants modify insect behavior, and there is a orientation preference for the insect and virus selection and the plant behavior. All three interactions will lead to the different mechanism of transmission. In non-persistent aphid transmitted viruses, specific changes in the host plant, such as suppression of jasmonic acid or signaling pathway is changed, the aphid behavior immediately will change in terms of landing and the feeding behavior. If say in whiteflies, we have seen manipulation feeding behavior of whiteflies, increase virus transmission. This process, again, there is change in the hormonal in the host, will also change the transmission, and which also is the genetic modification. This is also is demonstrated in our case, along with the Dr. Our student has demonstrated the whitefly preference in the susceptible compared to resistant types, where the preference is more towards feeding on susceptible lines rather than resistant lines. This is work was published along with the Kamala Jayanti. She has assisted. This type of work now is the where we need to study how the modifications are influencing in terms of resistance or in terms of host dissemination or host expansion. There are a color volatile organic compounds are the now current research, and mainly world is focusing on this. The volatile behavior and modification in the viruses which are leading to the quick attraction and changes. This has been shown in because of before contact, what is the volatile response? It is able to attract or not. Then post contact, after contacting, then there is a volatile change, which is again accepting the vector, assessing this for feeding, then it is spreading to niche. That is why there are indirect and direct manipulations have been now demonstrated in several combinations, especially in case of more studied in aphid transmitted viruses and to extent in thrips and white. So this is the type of work now we need to understand. It is not the insect is playing here important role. Virus is modifying. Once it reaches the host, it will trigger the host mechanisms to produce certain volatiles, which in turn attract the insect so that it's feeding on infected plant. Once it is acquired the virus, it is again modifying the behavior, which is again going to feed on healthy plant, not on infected plant again. Once an insect feeds on infected plant, it doesn't feed again on infected plant. It will go to the healthy plant and feed. Therefore, it is created an environment for survival. It wants that virus should be picked and should be transmitted into a new host. And this is been demonstrated in one aphid case, wherein the concentration of a virus is less, the virus is not attracting and virus is concentration is increased, it is able to generate a volatile which the aphid is attracted and it feeds, then it goes to the host again and feeds on the healthy host. So that is how these mechanisms are important. If all these mechanisms to understand, we need to apply proper diagnostics for detecting the viruses with the current technologies, the high throughput sequencing and ICAR. We are able to assess these type of modifications and relationships with the genomic level. So therefore we apply proper technologies for assessing these aspects. And as a influence, at the end, after as this basic mechanism we understand, we need to put into the practice for the managing these viruses. How we can interpret this of them? Most of the virus disease spread happens through the human influence. This is maybe because of to have a more food for us, we need to interfere. But without our, we also have a economy to increase and hence we diversify our trade and this thing. So the environment, the pathogen and host will pay one direction and human attracts this thing. When human interference is the migration in terms of carrying material and cultivation, monoculture or introduction of pathogens, new insect vectors. Therefore, this is all led to the interaction. If the disease triangle is one, whereas in case of viruses, it is not a disease triangle. The environment plays a critical in all are linked with the environment. Environment is more critical for these activities. Unlike other pathogens, the environment component, but here environment is a tripartite interaction between host . If you analyze these factors, some of the ecological and disease activity with biodiversity, the biological variations and the season, year are the host range, resistance and susceptibility are the factors which will change with the evolution. We need to, as a system approach, we need to go for virus management, not as a virus to be controlled. The one of it has been picked and transmitted by insect vectors. It is introduced, there is a signal transduction in the it cannot fail. Based on defending nature, the virus may be able resistance of reaction. And these are again the physiological expression of approach gene expressions, wherein this is expressed in every living cell and in the organism. Only we are currently identifying these factors. So to manage only we are able to successfully manage these viruses. So therefore, that is how we have to interfere, understand the basic mechanism, interfere those mechanisms. And developments made on plant disease surveillance, understanding tripartite interaction of host virus vector and plant disease modeling and decision support systems and integration of technologies to protect the national food supply. The convergence of technologies and networking the biological science, geospatial analytics, bioengineering, evolutionary needs to integrate and work together for a successful and development of a success. My talk. Thank you everyone for taking such a long time and your patience.
So much for giving excellent presentation and origin and evolution of this virus regarding the evolution of viruses and the complexities in even one variety dependent, and how 80% of this transmission is happening through insects. That is where the involvement of thank you so much for your excellent misconceptions regarding viruses. How best we can go with some points in that some points may come. Now I request please come forward and ask. You can send through chat also, or you can
Thank you very much, Dr. Sridhar, and but he has consolidated many things, which probably each one is a compiled, and it shows that he has got such a wonderful command over the subject and confidence to classification, to transmission, to the roles. My goodness, he is Dr. Krishna Reddy.
Thank you.
Sir, I have few questions, not questions that I read during my course of my humble queries that I have, if you permit me, sir.
Sure.
Sir, so do we have, are there any information regarding life forms and origin again?
Yeah, you have that statement. Viruses are universally present in all living forms. Virus is the only pathogen, I will say not pathogen, no other microbe exists in all living forms. But virus is the one which there in all living forms. You see in archaea, it is there. It is there in eukaryotic and prokaryotic. Whereas prokaryotic pathogens exist as a these things. And when we say when it's existing universally in all living forms, it is there where its expression is evident that is a disease. We see the pathogen, but it is there in all living form without. So it is not harming the host, and it is not, host is also supported by that. So that relations. One best I will show in our Indian condition only. If you see, jute is, and there is using for rotting and fiber extraction. In most of the cases, they juice is expresses. There is naturally one virus is present in that, and that virus, when you crush, that is a beneficial virus where it is used for commercial exploitation in Indian condition. Horticulturally, not viruses, viroids are used for dwarfing purpose. See, the viroid infected rootstocks have been used as a dwarfing rootstock. In fact, they are using a pathogen where we know certain cases dwarfing is induced by a virus, not by a genetic. These are few examples I would like. There are several examples. So virus is a friend and foe, both are, it is there.
Okay. Sir, few more questions. Can I continue?
Yeah, sure.
Accepted as binomial nomenclature and biological speciation. And species conservation. That is the three aspects kindly. One is biological. So under that circumstances, do viruses adopt the same kind of biological speciation? And
Committee on Systematics of Viruses. I am also a member for more than 15 years. And now we have introduced this classification. The binomial can implicate, at least where we can demonstrate that has been implicated. Now binomial species name is coming. Next version of ICTV, debating on this, there are both oppositions and supporting, and also discussions are happening how to bring on the way with the use of more genetic information. Unlike now we are using genomes as a biological species differentiation. We are insisting that host also to be included in the biological speciation, but there is a concern, it is still a sure.
Please, because in such discussions only I can approach Krishna Reddy. Otherwise, we joke, we casually talk, but this is something professional talk. Let's. So I'll take this opportunity to ask you one or two more questions. Does single and double partite and satellite types have any and translation?
Yes, now
now, because till we have come to know about siRNA, miRNA, we were not knowing post-translation extra inheritance matter influencing the phenotyping or the characters of the or transmission of or of this single double partite, then satellite types. There is a variation in the modification because we are using that information for the benefit of the agriculture or these kind of newer particle systems that are evolving from the viruses can be transcription translation mechanism changes.
Yes, as per now, the research carried on DNA satellites, the beta satellites are
Voice breaking.
if the beta satellite is associated symptoms,
No transmission, sir.
host satellites. And now people are also attempting the modification in the beta satellites would lead to the role is not proven, and delta satellites is now only discovered, association only recorded, but role is not at assigned. So unless the role is we know, then only the how to intervene will come to the later stage. Whereas in RNA satellites, it has been demonstrated interference. RNA satellites have been used in RNA approach is used RNA satellites for modification behavior of the certain RNA viruses.
Right, sir. One last. Viruses are deciding the type of host. Does it mean that if a white fly is infecting with the bigomovirus for a particular host, that means that it is it mean that there is a kind of natural elimination of supremacy is taking place?
Yeah, ecosystem once they establish, other virus will not come. That system happens in case of bigomoviruses. If they that other insect, aphids or others which are feeding are not attacked. Whereas in aphid, feeding on that. So you have a aphid transmitted virus present in the host, and you have a whitefly transmitted virus is also present in that. Whereas in whitefly transmitted, the whitefly transmitted virus presence in the beginning, other insect transmitted viruses are not prevailing that much. But if aphid transmitted virus a specific role, whereas aphid transmitted and thrips transmitted a generalistic approach. So their possibility of both are coming. Patterns. We need to understand more whether in all cases it happens or not.
Krishna Kumar, this through you, all of us got benefited. These are the actually common doubts, and thanks for. And there is a one question from Dr. Sirila Chalam from NBPGR. Congratulating you for excellent presentation. Her query is: seed transmission of begomoviruses will have trade implications. how many viruses are
of trade implication only I was mentioning. It is a genotype dependent. So is transmitting in certain legumes, and tomato leaf curl new delhi virus is transmitting. In my slides also I shown in the field bean. So far we tested several samples of tomato leaf curl. The monopartite viruses, we could not detect the seed transmission. Similarly, in okra also, monopartite viruses we tested, we could not detect. But there is a time factor. What I am saying, when we tested, one example I wanted to quote here. When we tested in okra bigomoviruses, yellowwing mosaic and leaf curl, in 2008 and 10, okra Asian leaf curl virus we could detect in the seed coat. So there it may come. At present, we can define published viruses, we
That's
can
right.
notify
Yes.
whether virus present or not. But it should not be applicable uniformly to all the host. It is the host.
Thank you, Dr. Reddy.
Yeah,
Yeah, that's
you are
Celia.
more important. You have to see this implication. Most of the seed transmission part as a trade barrier.
Yeah, I wanted to write that.
It's talk, not in the publication.
Yeah, thank you.
Yeah, thank you. Next one is Dr. If so, how to detect and apply for the management of their specific insect vectors. Here, Dr. Celia has replied one small example also. In addition to that, you can share. Plum pox virus is known to produce vivosis, and dogs are used to detect these viruses in fields in USA. That is that Celia's means answer. You can go further. Sir.
In turnip mosaic virus in crucifers, that was the first virus demonstrated volatile protection in crucifers. In relation to aphid attraction, it was demonstrated. In now, the work is now initiated. That is my talk mainly was interested. My way of approach here is to instigate more youngsters to work on this. We need to work on this area. Volatile production is the main concentration and the insect virus host interactions. We need to study more virus host combinations. Now we need to study these things. Yeah, there are excellent facilities available, several institutes. That is only HPLC and GLC system required. And if anybody, I think our scientist, Dr. Kamala Jayanti is there working on volatiles. She can guide and also collaborate. People can establish working on volatiles in relation to these aspects.
Yeah, next another is from ICAR-IIHR Bikaner. Thanking you for giving in such informative talk. His query is, as evolution is a continuous process under the present scenario of these viruses, what we should do in for the management of these viruses in future?
My last slide is that only. We have to interfere in different processes, the insect behavior and the modifications. Simple to say in virus modifications, management. The simple border cropping is itself is the one which prevents the feeding behavior change in the behavior of the insect. So it looks not a science and any chemical application. One, it is not 100% will stop, but there is a slight modification in the insect behavior, which is again is helpful. Similarly, reflective mulches will also modify the behavior of the insect vectors. And the spray of, now I am saying the microbes, we use microbes for fungal and bacterial diseases. I am recommending use even viruses. They don't directly stop virus multiplication, but they in turn influence the host to modify behavior. Host modified behavior also is the one which can be influenced for disease management.
Okay, thank you, sir. Anybody else would like to ask any further clarifications? Yeah.
Namaste, sir. Namaste, sir.
Yeah, please go ahead.
Chandrasekhar.
Myself, Dr. V. Chandrasekhar, sir, from RR Sankapalli.
Okay, please go ahead.
First of all, I must congratulate Dr. Krishna Reddy, sir, for giving such a elaborate lecture and very insightful lecture for all our audience, sir. Thank you very much, sir, for giving such a great lecture. My question is, sir, first of all, what is the necessity that viruses means? What make them to evolve, sir? Because in our cases, multicellular organisms, there is a lot of competition among us is existing. But viruses are such a tiny and they are simply nucleic acid. But still, they are very rapidly emerging and evolving. What is the necessity that making them to evolve, sir? That is my first question, sir.
Yeah, my beginning. I understood, Chandrasekhar. My answer is, in the beginning, I mentioned I consider viruses as a living. Okay, if you consider as a living, like any living organism tries to compete in the ecosystem, tries to dominate and exist. Their urge is to exist.
But here there is no competition among themselves.
No,
sir.
yeah, competition is there. See, competition is there because their survival. See, you are, one example I will tell you, you are spraying indiscriminately on insects. Insects are died, but their vehicles are not there. How do they survive? One way, that is one. Second, we also change the hosts. We also breeding the different hosts and to modify, and so in terms of yield or enhancement, whatever it is, the genetic nature of host plant we are changing. So when there is a pressure on these things, definitely they have a pressure to survival.
Yes, sir.
That is the survival mechanism only. They are not interested to dominate the world, but they are survival mechanism. Best example I will say, in our medical system, see, we have eradicated smallpox. Do you agree that we curtail and it is same way cholera? We no more we heard cholera, but we have now corona. Why? Because if you suppress something, something else will come out.
Yes, sir. Yes, sir.
We are suppressing. We say they don't have competition, but they have competition because our behavior is changing their behavior.
Yes, sir. Yes.
Yeah, that is how it
Sir,
is.
My second question, sir, in our biological or metabolic system also, lot of gene splicing and all such mechanisms are going on, sir. So every moment there's transposons and everything are evolving. Is there any likely chances that they can become as a viruses, sir? Because the maximum portion in our genetic constitution is of brain matter. I mean to say that it is non-coding region. So while splicing, is there any possibility that such kind of regions can become as a viruses?
Yeah, that is what symbiogenic theory is, that only. I mentioned now one symbiogenic theory, that is that only. In the host, integration genomes are there in the host. When there is a stress, they becomes independent events and then becomes a virus part. It is there now.
Is any such examples are there, sir? In our case, at least in human being.
Banana streak virus and also rice tungro virus, all these are all that such viruses exist, splitting only.
Okay.
During stress, it will become virus particle. Otherwise, integrated virus will be there. Even in human and animal also certain viruses are there, but exactly I will not able to tell now because I not remembering all at this moment.
Yes, sir. Yes, sir. I do. Sir, another important question, sir. In one of your slide, you are saying that, of course, highest evolving organism is virus. Then followed by you mentioned as fungi, sir. To my surprise, actually bacteria is having more chance for evaluation, sir, than
No,
evolving than
no. No. My slide is not on that. Slide is not on evolution. It is the number incidents. Incidents.
member.
Number of viruses, diseases recording. I'm
No,
not saying
no, sir.
evolution. I didn't mention there. I mentioned the population. Now number of viruses are increasing. Earlier, fungal are the more number in terms of pathogens. Now fungal is dominated by viruses. That is the number of viruses reported is increasing. That is what my meaning, not a evolution.
Otherwise, bacteria is having more scope for evolving, na, sir. That is for sure,
No.
na, sir.
I am referring to only plant. In plant, bacteria is less only. Number of bacteria in plants are less, though they cause maximum damage, but the number of bacterial species in the plants are less only.
Yes, sir.
You are talking as a microbe, that evolution. Yeah, I do agree. Evolutionary point of view, they are more have a dynamism, but number of
Yes.
bacterial species affecting plants are less only.
Yes. Okay, sir. Thank you, sir. Thank you very much.
Okay, thank you. Anybody else?
Chart anyone?
Yeah, anybody else? Dr. Vamana?
Yes, sir. Sir,
Yeah.
good afternoon, sir. It is excellent presentation, sir. We enlightened with your presentation, and we learned one very important thing. Many viruses are, we are generally saying seed-borne. It was earlier literature also, it was fixed. But today I came to learn that it may vary. Even it is seed transmissible, even in another case it is insect transmissible. Like that, really we came to know from your lecture, sir, today. But one thing in Rayalaseema districts, as you know very well, sir, Parthenium, it is pollen-borne virus, TSV is there. But since 20 years before itself we reported, but farmers are unable to control the Parthenium weeds. And it is spreading like anything, and every season we are getting, sir. So for this, we are unable to control this virus, especially Rayalaseema district, sir. In future, what will happen also, we don't know, sir. If there is anything, please interact, sir.
Tobacco streak, you know, at least no, but that is the what I said in the ecosystem also plays an important role. See, in that ecosystem, it is not the Parthenium alone is playing. It is the Parthenium pollen is carried by insects. So the insects are the ones which are the main vehicles.
Yes, sir.
So we are looking only on Parthenium. We need to look at the insects carrying the pollen. That if you determine which are the insects which are carrying pollen. If we are able to manage those insects or reduce those insects, certainly we can bring down. In addition to Parthenium, we need to bring down the insect population in that region. Same example I can quote. See, you are our student, both your student and my student also
Yes,
here, you know, that tobacco streak.
Yes, sir.
Parthenium as a source, but in Karnataka, Parthenium is so much, it is not a source for tobacco streak virus. We could not detect much what you are finding in Rayalaseema area.
Yes.
So there is a ecosystem also plays an important role.
Sir, this tobacco streak
Thank you,
virus
sir.
is not a seed transmitted, correct, Nathan?
It is seed transmitted in certain hosts.
Okay, there was a query from in one of the meeting.
Yeah, I'm telling no.
Yeah,
Tobacco streak virus is a seed transmitted in cucurbits.
cucurbits. Okay, okay.
Transmitted in legumes.
Okay.
And
In
even
this-
in sunflower, it is not seed transmitted even in sunflower.
Okay.
Dr. Reddy,
Yes, sir. Thank you, sir.
Dr. Krishna Reddy, you say that pollen carrying insects have to be reduced, but many of the pollinators do carry the pollen. And
I mentioned
a
that
paradoxical
ecosystem
situation.
approach. No, not honeybees and all these things. ones which feed on them. See, honeybees also carry pollen, but they don't feed on the plant. They are only pollen collectors, nectar collectors. So during pollination time, it is what I am seeing, referring to thrips especially. They are the ones which also carry as also feed. So that type of thrips, not the honeybees and all.
Yeah, there is a query, sir, from ICAR-IIHR Bikaner. They had detected one Begoma virus in seed in first year using specific primers, but second year unable to detect using the same primers and PCR program. They are asking for your comments. Year to year change.
No, if that's my answer is earlier, if you listen carefully my talk, if the seed detection, was it the same genotype you are not able to detect in the second year, or it is a different genotype? If it is different genotype, you will not get it, because that genotype, it is a seed borne. It is not necessarily that you should be seed borne in this variety. That is the primary difference. The second one is the primers what you have used. Maybe one set of primer, you need to use more sets, you are able to detect the virus if it is really present.
Okay, sir. Another is tobacco streak virus in okra is seed transmitted?
No, it is not seed transmitted in okra. We have tested that, but we could not detect in the okra seed.
Okay, thank you, sir. Anybody else? There were a lot of questions, so many things we could learn today. And last call, if anybody is having any questions, you can go ahead. Otherwise, yeah, from we are coming to the end of the session. Once again, we profusely thank you, sir, Dr. Krishna Reddy, sir, for accepting this one in a short notice and giving a wonderful lecture on evolution of these viruses and understanding of these viruses, how it is happening, how it is bettering year after year. Thank you so much for sharing your experiences, last 30 years experiences. Thank you so much from our AAPMHE and IHR for your excellent lecture. I also thank Dr. Nagesh, Dr. TVK Singh, Dr. Celia Chalam, Dr. Madhu Reddy, and many others who could join in this talk, and also making value addition by participating in this interaction. I thank one and all, and let us meet next month with another topic related to plant protection. Till then, I say thank you. Yeah. Tell
Sridhar, I would like to thank you for giving this scope to the idea.
Yeah.
My only interest now, and also our colleagues in the division, should work more on volatile and interaction of insects and viruses, so that we give more priority for our research in the division. And that's the main thing. And you will have a lot of links with other institutions, other universities.
Right.
The area we need to promote. That is main idea I wanted to have. And we have excellent facilities and expertise also. Kamala Jain, I don't know whether she joined in this or not.
Yeah,
she is there. Our plant pathology colleagues as well.
Yeah.
And they should take now more role in this direction, so that everybody will ask, what is the control for virus?
Yeah.
Unless we understand the basic mechanism, it is difficult to interfere with the system.
Right.
Sir, I am there, sir. I am hearing your lecture. Yeah, definitely, sir. I will take forward. Definitely, I will be in touch with you also for suggestions and guidance.
And now more of youngsters are there in the virus, so you can have all of them put into this direction and have more productive work.
Yes, sir. Yes, sir. Definitely.
Okay. Thank you, Dr. Kamala Jayanti. Thank you, sir. With this, thanking you once again, we are closing this session. I thank one and all who joined in this session and participated actively. Thank you so much. Let us meet next month with a new topic. Bye. I'm closing the session. Thank you.